Plasmids are so useful because of their flexibility. You can insert nearly any gene you want. Maybe a GFP gene to illuminate your work, or you're trying to study the effects of a specific mutation on cells. But how does the plasmid express the gene of interest? For an answer to that, let's talk about the promoter region. The promoter region is the element of the plasmid responsible for initiating the transcription of your insert gene into RNA. This means it gives your plasmid the green light to start RNA transcription. When we talk about the promoter region, we're talking about two distinct parts of the plasmid: the promoter and its DNA regulatory sequences. The promoter is the site where the RNA polymerase binds to the DNA, enabling transcription. The DNA regulatory sequence controls are the mechanisms that allow the gene of interest to be expressed. We'll talk about DNA regulatory sequences a little later. For now, let's focus on the promoter. Promoters are typically between 100 and 1,000 base pairs long and are found upstream of the gene of interest. It's these sequences that control the binding of the RNA polymerase and transcription factors. As the RNA polymerase moves along the DNA strand, it creates a new strand of mRNA. This mRNA contains the instructions that tell the insert gene to express. In addition to RNA polymerase, the other regulatory elements influence when transcription occurs. One example of a regulatory element is an enhancer. These short regions of DNA are upstream or downstream of the promoter site, so they need to be looped into the transcription process. Enhancers bind to the promoter to increase transcription of the gene.
Transcription enhanced, time to dance! Alright, woo!
Another regulatory factor you may encounter is a silencer. Silencers are regions of DNA that elements called repressors can bind. When a repressor binds to a silencer, it inhibits the RNA polymerase and no mRNA is produced. This essentially shuts the gene off.
Where'd the light go?
Another regulatory factor you may encounter is an insulator. Insulators act as a buffer between the promoter and other elements like enhancers by binding to the promoter so the enhancer cannot.
Come on, let me in there. Come on, let me in. Come on, I'm on the list. Come on, you gotta let me in there.
Different RNA polymerases require different promoters. You'll need to keep this in mind when you're selecting the promoter for your experiments. Additionally, there are several other considerations to make when choosing a promoter. Some promoters are active all of the time. This may be useful when using something like GFP, where you'll want the cell of interest to always be fluorescing. Other promoters are more carefully controlled. Some may only act in certain tissues. Others only act at certain times. While others can be turned on or off by different factors, such as specific sugars, light, or even heat. Sometimes transcription doesn't go as planned, and leakiness can occur. Leakiness is when transcription occurs when it's not supposed to. For example, when a repressor protein is bound to a silencer. In nature, this is common and generally won't cause any issues. But it could spell doom for your research. For example, if the insert gene is toxic, leaky transcription could lead to the cell being poisoned, ruining your time-consuming research. One way to ensure this doesn't happen is by using a synthetic promoter. These promoters are not naturally occurring, but are constructed in a lab to achieve optimal regulation. With all of that information, it's time for you to head into the lab and express some genes. We hope you enjoyed this video in our Plasmid 101 series. Don't forget to subscribe to the Addgene channel to catch our newest videos, and click share to help spread the word. And as always, visit blog.addgene.org for the latest plasmid info. Addgene, a better way to share science.